Dear students, welcome to SETI Botany classes. In this video, we will study about restriction endonucleases, what are different type of restriction endonucleases, and how do they cut DNA molecule. Now, these are enzymes which are highly specific. They will recognize DNA at specific places and usually isolated from various types of bacteria because they can cut DNA molecule. So they are also called as molecular scissor. They cut DNA molecule within the living organism. They are also called as biological scissor. So specific sequences which are cut by restriction enzymes are called as recognition sequences. Now these sequences may be made up of 4 to 6 base pairs in length. So we can define restriction endonucleases are enzymes that cleave DNA molecules at or near specific nucleotide sequences. So they are highly specific sequences where these enzymes will act because they can cut within the DNA molecule. They are called as restriction endonucleases. Now, when they cut DNA molecule, two kinds of ends may be generated, blunt ends and staggered ends. So we can see here double stranded DNA. This is also double stranded DNA. This is running from 5 prime to 3 prime direction. This is from 5 prime to 3 prime direction. Here also 5 prime to 3 prime and then 5 prime to 3 prime. The sequence is C, C, G, G. And in the opposite strand, same sequence is there, C, C, G, G. Such sequences which are same on the opposite strands of DNA are called as palindromic sequences. They are mirror image of each other. Here also we can see the sequence GAATTC from 5 prime to 3 prime direction, and in the opposite strand from 5 prime to 3 prime direction, sequence will be same GAATTC. So again, this is palindromic sequences. So restriction enzyme can act on highly specific sequences, and these sequences may be palindromic sequences. And when they cut DNA molecule, this is the site for restriction enzyme. This is arrow is representing site for cutting the restriction enzyme. So here when they cut the DNA molecule, two blunt ends are generated. But here when cut is there, sticky ends are generated. We can see opposite to TTAA, no base pairs are there. And here also opposite to AATT, no base pairs are there. So these ends they are called as sticky ends, staggered ends, or cohesive ends. So two kinds of ends are generated when restriction enzyme act on the DNA molecule, called blunt ends or staggered ends. Now, there are three types of restriction endonucleases: restriction endonuclease 1, restriction endonuclease 2, and restriction endonuclease 3. These enzymes are very important for genetic engineering. Now, type 1 enzyme is highly complex and its recognition sequence is made up of 15 nucleotide base pair in length on the DNA molecule. So, this enzyme can bind to the DNA molecule, but this will act on the random site for cutting the DNA molecule. That may be about 1,000 base pair away from the 5 prime end of the recognition sequence. Important type 1 restriction enzymes are EcoK, EcoB. How naming of the restriction enzyme is done? This is called as nomenclature of restriction enzyme. This we will discuss in the next video. Now, this is type 2 restriction enzyme, which is actually involved in the genetic engineering. Uh, this can bind to the DNA and cut the DNA molecule within the recognition sequence, but for its activity requires magnesium ions. And examples of type 2 restriction enzymes are HIND3. This has been discovered from Haemophilus influenza. This is the name of the bacteria. So this is the actual enzyme which is involved in the genetic engineering. Now, this is the type 3 enzyme, which require energy for its activity in the form of adenosine triphosphate, that is ATP. This can cut the DNA, can also add methyl group. So this is called as modification enzyme. This will again bind to the specific site, but this will add the methyl group. So actually, this is not involved in the genetic engineering. Examples of type 3 restriction enzymes are EcoP1, EcoP15, HIN-F3 enzyme. So these are different examples of type 3 restriction enzyme. Thank you for watching the video. If you like my video, please like, share, and subscribe.